So today I'm going to split the talks into two sections. The first section will be covering, as Laura mentioned, detection of horse meat. And the second section will be covering the use of LCMS for the detection of allergens in food. And so I'd like to start off with the horse meat. I just want to sort of set the scene. Basically, about a couple of months ago, as people probably realised, that there was a huge food scandal in the UK where horse meat was found in food which was not labelled horse meat, and also it was found at actually levels up to nearly 50 to 100%. This proved not only label issues but health issues, and there was a huge food recall in the UK which cost the food industry millions of pounds. And when this happened, I had discussions internally about whether we could use LCMS to develop a method not only for detection of banned vet residue which are used in the racing of horses, but also in the detection of speciation or horse meat itself. And this is what drove me to the slides and data that I'll show today in this webinar. Sort of the agenda for the first talk is basically a brief introduction about the history and the impact and the background to this piece of work. The second one will cover some of the theory behind protein detection by LCMS. This is really based on the academic workflows and how you can apply these to routine food testing. And then I would like to just discuss what the potential of micro-LC is in this area, which is a new LC technique which is probably around for a couple of years, but it's now become commercially available with specific micro-LC instruments on the market. And finally, I'd like to discuss some conclusions I can make from this work, which is the start of a series of applications that I'll be working on in sort of meat speciation. So looking at the theory around meat speciation, basically in meat speciation what you're trying to do is identify protein markers for the species. In LCMS what you do is you first of all identify the protein marker. You then generate the fragments for the protein by breaking up, typically by enzyme digestion. Most people use trypsin, or they can use other enzymes. Once you've broken up the protein, and then what you generate is a list of peptides. The peptides, of course, produce a Q1 mass, which is their parent mass. They then can fragment them in a sort of a triple quad gas cell, and this produces fragmentines, which are characteristic of the peptide. This also, you have the ability in Q-trap technology, which is sold by AB SCIEX, to generate full scan product data at the same time as acquiring this sort of MRM or Q1, Q3 transitions. And this product line data can give you full sequence coverage for the peptide as well, which gives you additional points of confirmation and additionally confirms the presence of that peptide and therefore that protein in the sample. And then it sort of loops around again, and then you repeat this for a series of protein markers and peptide markers. So basically what you do is you identify the species you want to detect in your sample. And in this case it was unprocessed meat, so it was raw meat. So for example, you go to horse, so you look in the literature, find the proteins related to horse, and then you would do what's called an in silico digest. This is a sort of a theoretical digest of the protein with the enzyme you plan to use. This will give you a sequence of peptides, and then from these peptides you can then go back to the literature and search to make sure that they are unique to the species and unique to the protein. This is really important because what you don't want is to have false positives with species identification. So you really want to make sure that these peptides are unique. And then you run it through what we call this Midas workflow. So once you've decided on your list of unique peptides, you then plug them into the MS by basically, because the in silico digest would also give you theoretical peptide transitions, or MSMS, for these peptides. You can then put them into a standard C18 method, so a method run on a C18 peptide column by reverse phase HPLC and positive electrospray. You then basically extract your meat and see if you can detect the peptides and confirm them by the presence of this full scan product ion data, which is acquired in the Q-trap. This approach is called MRM-initiated detection and sequencing, or the Midas workflow. So you have a series of these MRMs which correspond to a series of peptides for the target protein, which are unique to the species. You then look for all these, and as soon as you find them, it triggers the production of a product ion scan, which then gives you the sequence data. So not only do you have the MRM and the MRM ratios for each of the peptides, but also you have the sequence data from the product ion scan to give additional confirmation. And this is really quite unique compared to other techniques like ELISA, where ELISA just picks up the one section of the protein which binds to the antibody, and so it only has one point of confirmation, one point of detection. With MSMS and LCMS, you can have multiple peptides and multiple points of confirmation, which gives you far more confidence in your results and removes a lot of the issues of false positives that sometimes you can get with ELISA. So basically, going back to how to develop the methods, you really, before you do this, need a source of either the pure protein or the species. So you need to, in my case, for some of these, go into a butcher which would single source the meat for me, or, for example, in horse, what I did there is actually look at Sigma Aldrich, and one of the proteins which is found in horse meat, horse myoglobin, was commercially available. I just went and bought that directly from Sigma Aldrich. I then ran a discovery experiment, as I explained, with these target peptides. I then double-checked once I found them to confirm the sequence of these peptides matched the peptide sequence in the theory for these peptides to confirm their presence. And then I double-checked all these sequences against the literature to confirm they were unique. So the peptides I chose for horse meat were unique to horse and the horse species, and were not found in, for example, beef, lamb, pork, chicken, or in some cases even rats, because rats can be an issue sometimes in certain countries in meat manufacturing. The other thing you need to think about as well, if you move the assay into processed meats, is to make sure you eliminate peptides which can undergo some modifications in processing. So what I additionally went and did was looked at the sequences and determined whether or not they would be affected by processing, for example, the Maillard reaction, or modification of the methionine residues. So you need to make sure you remove these if you specifically want to look for processed meat, because these can be changed in processing and therefore vanish in processing. And this again is sometimes seen in ELISA, where some of the markers disappear when the actual samples are processed. And this is seen specifically in allergen detection. So at that point then I designed my MSMS method, and the next thing I wanted to do was just to try and set up a full LCMS screening method, and I went to use micro-LC. Now, one of the things micro-LC gives you is that it means that you're running samples at a lot lower flow rate, which gives you sensitivity increases. So, if you look at discovery proteomics, which is a lot of this work's done in, for example, academia, in shotgun proteomics, for example, the ETH, and they often use nanospray, because nanospray is well known to give you sensitivity increases in protein and peptide ID. Well, micro-LC sits in between nanospray and high flow systems. High flow, I mean a flow range round about a couple of hundred microliters per minute. Micro-LC actually runs round about 10 to 40 microliters per minute. So it is a factor of 10 to 20 below the flow rates of normal high flow LC. But it gives you sensitivity improvements because of the efficiency of evaporation you get at these lower flow rates and the efficiency of ionization you get. So that's one of the potentials of micro-LC. For a micro-LC system, what you actually need is to make sure that you have something which is like a standard LC, is capable of giving reproducible gradients. You need something which is capable of using these narrow bore columns, because these are essential for micro bore LC to have fast chromatography, and something with very little delay volumes. And what the Exigent Expert Micro LC200 system gives you is all this. So it gives you gradient delay volumes of less than a couple of microliters. You can do injections up to 10 microliters, just as far as I've gone, and you have a column oven which fits very, very close to the source to reduce dead volume, which goes up to 80 degrees C. So it's uniquely positioned for running micro-LC flow rates. What AB SCIEX has done as well is to modify the source for these flow rates. One of the things you have to be conscious of when you're using low flow rates is band broadening of the peaks due to dead volume emissions post-column. And what AB SCIEX have done is they've generated a set of new capillaries, which is electrodes which fit into the source probe, which are really low dead volume, which mean that you get reduced band broadening and very sharp peaks, so you don't lose your resolution after the column. And these are very simple to fit, so basically just change the capillary in the probe, and it takes about a couple of minutes to do. And it's an add-on. It means you can use existing turbo V sources for this work. And this is what I did here. I just simply changed the probe for the 25 micron ID probe and ran the analysis. This just shows you an example of the sensitivity gains you can get. This is a screenshot of a high flow compared to micro LC flow of some peptides. This is not meat, this is milk. But you're seeing factors of 3 to 10-fold increase in signal and signal to noise for the peptides when you're running at 20 microliters per minute compared to 250 microliters per minute. So you get a consequent increase in sensitivity running at these low flow rates. This is one of the reasons why I chose to use micro LC rather than high flow LC. So the method I chose to develop and run the meat speciation samples, which were actually horse meat samples and meat samples, was based on micro LC. I used a core shell halo C18 column and a gradient from 2 to 98% acetonitrile, which is sort of containing 0.1% formic acid. And this is sort of typical mobile phases you would use in protein research. The column oven was set to 40 degrees C, just simply so I can maintain an accurate temperature above room temperature. And I used 10 microliter injection volume, which is again sort of typical of the sort of injection volume you would use for protein peptide analysis. Looking at the extraction is sort of developed on previous experience running with allergens. Basically, I took a food product, in this case meat. I extracted it with buffer, and then I modified the proteins to enable them for digestion. So what you have to do when you're doing this sort of work is you have to reduce and alkylate your proteins prior to digestion. This opens up the protein structure and means the enzyme works more effectively on digesting the protein. This was done in this case by using TSEP and MMTS, which is TSEP is a sort of a sort of reducing agent. MMTS, which is a sulfonate, is an alkylating reagent. This whole process took about an hour and a half to reduce and alkylate, and an hour to extract your proteins from your meat. And then we just did an hour tryptic digestion, not 14 hours as this slide says, a typo there. And then we did offline SPE to concentrate up our peptides, and then we analyzed them on the micro C system. This sort of approach, sample prep, took about a day to do. So you start off in the morning, you can get it done in about 4 or 5 hours from the sample to result. Oops, let's go back to the different slides. This is just a slide showing the sort of comparison of different meat sources. So what I did, once I developed my MRMs, my target peptide list for horse, I then got some reference material of beef and pork. This is from LGC. The horse reference material is from LGC as well. And I got a sample of lamb from a local butcher. I then extracted all these samples and then compared to make sure I had no false positives for the horse peptides in either lamb, beef or pork. And you can quite clearly see that the horse peptides are easily distinguishable, and they're not found in any of the other species. At the moment we're working on producing markers for other species, but so there were additional markers present in here for beef, but not so much for lamb and pork, but the horse meat was specific for horse. I then actually got hold of some reference material, again from LGC. This was run on a PT trial, which is performance trial for ELISA and PCR methods for horse meat. And what they were looking at was looking at detection limits and a sort of semi-quantitative approach to see how low current methodology could go for the contamination of horse in meat-based products, for example, meatballs or lasagna. And this is what I got hold of. These are basically blended samples, so they weren't processed foods. They were actually blended samples of meat where beef had been spiked in with horse at certain levels. the current methodology by ELISA and PCR is normally detection down between 1 and 5%. So my goal in developing the method here was to get down to those levels with multiple markers, not a single marker. And what you can see here from real extractions of this horse meat sample is that we could see down to and pick up 3 peptide markers down to 1% horse meat in beef. And this is not the sort of the vet residues, this is actually the protein markers using this sort of workflow. And this was based on a 5500 Q-Trap platform, not our top end 6500 Q-Trap platform. So I know if we need to get lower, there's a potential of getting lower detection limits using the more higher end Q-Trap system from ourselves. But this clearly shows that the method for detecting one species horse in beef can detect 1% horse meat in beef using LCMS for the peptide markers. The other question I had at this point was, could I then use a similar approach to detect the vet residues in the same analysis? A lot of methods currently out there for detecting horse meat by LCMS weren't looking at the peptides at the time, they were looking at the vet residue phenylbutazone. Phenylbutazone is used as a painkiller for horses, for racing horses, and for horses using show jumping. And it's actually a banned veterinary drug and should not be used for human consumption, so it's illegal to be present in meat. And people were using the detection of phenylbutazone as a marker for horse meat rather than the detection of peptides, which I'll show here. So I then decided to see if I could combine the method by adding phenylbutazone transitions to the LCMS method. So this is what I did. Before I do that, Lauren, do you want to just ask the polling question before we move on?
Yes, thank you. So we have a polling question for the audience. Actually, we have 2 polling questions for the audience, and you can enter in your answers right there on your screen. The first question is, what other meat or tissue species would you be interested in testing for by LCMS-MS? And you can just type in your answer right in the box. And the second question is, what levels of contamination of assorted meat species would you like to detect? And your choices being less than 1%, 1 to 5%, 5 to 20%, more than 20%, or you don't want to issue a vote. So again, what other meat or tissue species would you be interested in testing for by LCMS-MS? And what levels of contamination of assorted meat species would you like to detect? And as we broadcast the results, for the levels of contamination, we can see that we have anywhere between 1 to 5% or less than 5%, or excuse me, less than 1% are the concentrations you would be looking to detect for assorted meat species. And when we look at the other answers for what other meat or tissue species would you be interested in, we see some comments on pork, beef, halal meat, and we also see seafood products, shrimp or prawn, for people who have allergies. So Steve, do you have any comments on those results?
Yeah, I think the levels sort of probably match where I'm here from the industry. About a 1% is typically demanded as a sort of LOD. So that's why I designed the method around the 1% detection limit. Regards to speciation, that sort of again confirms what I'm hearing from other people in the industry I speak to around especially pork and beef. And we are developing markers for those. This is just the first stage in a process where we're developing future methods for meat speciation, and I'm working with academic researchers to increase this list. What interesting for me is about halal. Again, halal is a very interesting area. Of course, pork is part of this whole question, but there's other markers for products derived from species like collagen, which is used in gelatin, which is important for halal, and we're also looking into that as well. So we are looking into these areas to see if we can produce methods for detecting things like gelatin speciation, which is important in the confectionery industry. So it's good to see that people are passing comments which is round about the area we're thinking of carrying on this research into. So that's good.
Great, thanks, and thanks to everyone in the audience for submitting your answers. So we'll go back to the presentation and let Steve continue.
Yeah, so before I come to the audience poll, I was mentioning the issue of phenylbutazone, or bute as it's called as well. You've seen it in the news. This has been used as markers for horses in meat, and the reason is because often when you don't trace a horse, some horses are bred for eating in certain countries. It's not just this is the UK where they're banned, but in other countries they're allowed to be eaten. But if a horse has not been bred for consumption, it's been used in horse racing. This is when phenylbutazone has been used, and this is where they can get in the food chain. This is why the labelling is so important in the UK to ban horse. This is why phenylbutazone has been used as a marker for horse meat. And the question I had at the beginning of this work was, could I combine peptide marker species as well as vet residue analysis in the same approach, in exactly the same method without any alteration? This is what I tried. And here you can see just a shot. This is really a first pass trying at this. Is very early results of looking at phenylbutazone in spiked into horse meat compared to the horse meat blank, and also compared to the standard. So if you look at the results, you can quite clearly see a 2.5 ppb standard is quite clearly detected using the same method. So basically this is using the same extraction protocol, so I haven't changed anything, and the same LC conditions of the speciation. So everything's the same. I've just added the MRM conditions of phenylbutazone into the method. And you can see here as well, when I spike in the 2.5 ppb, you can still detect it in the horse meat. So it clearly shows that you can combine vet residue, banned drug residue testing, and speciation into the same workflow without any modification to the sample prep, which is really, I think, is a huge improvement in throughput. So for people who want to test for this, you have multiple points of confirmation. Now you can test for the vet residues as well as the speciation peptides in the same analysis, and this is unique to LC-MS. You can't do this by PCR or ELISA formats. They can't test for the phenylbutazone at the same time as detecting horse. This is something unique to the technology of LC-MS, which I've shown here. The ultimate sensitivity hasn't yet been tested for the phenylbutazone in this method, because the point of doing this work, I couldn't get hold of the Butte standard. It was sold out in Sigma just because all the labs were testing for it. So I actually used encapsulated horse medicine from a colleague who supplied me a sachet to extract and run. So I'm not quite sure of the actual final concentration of my phenylbutazone I spiked into the horse meat. So I can probably get a lot lower than I actually show in this method. Lauren, the second poll question, please.
Yeah, so one more very quick poll question for everyone. The question is it necessary for you to detect and identify both meat species and veterinary drug residues in a single analysis? And your choices are yes, it's essential to analyze for both the species and the veterinary drugs in the single analysis. Maybe it's a nice feature of the method but not necessary for your lab. Or no, it's not essential to analyze for both species and veterinary drugs in a single analysis. So again, is it necessary for you to detect and identify both meat species and veterinary drug residues in a single analysis? And if we look at the results, we see that the majority of you are saying it's a mix between yes and maybe. So yes, you think it's essential, or maybe it's a nice feature, but no one said no, it's not essential. So it looks like the audience does feel that having this mixture of analysis of the species and vet drugs in a single injection is a benefit.
Okay, that's excellent to hear. This is very early results, and we can always add more drugs, vet residues into this method, because we're using a sort of standard SPE protocol, so which tends to trap a lot of these vet residues on the column. So, and this will help us get down to detections for vet residues as well. So it's good to know that we're working in the right direction here for developing solutions for customers. So I'd just like to conclude this section of the talk with some conclusions I can achieve from or get from this work. I think now we can see that from the results I've shown today that LC-MS has the potential to offer rapid, robust, and sensitive assay, very specific assays for meat speciation. And you can see here from the horse meat that you can get multiple markers which are unavailable from other types of detection using LC-MS. And the sensitivities you get are equal or even better in some instances, or potentially better than current technologies like ELISA and PCR. At the moment, the test is still qualitative. We haven't really looked at the quantitative side of this. I'll cover quantitation in this sort of area more in the allergen talk following this. But as I was showing, you can get down to the limits. I think one thing, just a note of caution when developing these assays, you have to be careful to choose specific and unique peptides for the species when you do this. This is just a point of care for anyone who's thinking about looking into the area. And I think one thing which is unique about LC-MS in this area is the ability to combine the peptide and species detection from the proteins as well as the vet residue detection all in the same sample prep and analysis, and this simply can't be done by other types of technology. Before I finish this talk, I would just like to acknowledge the University of Münster in Germany. They've been consulting with me on this work, and they're actually working with us on developing more species markers, and they've helped in this work. So I'd like to acknowledge Professor Hans Humph for his support and help in this project. And I'd like to just thank you for your attention.